Good day, scientists, and you're welcome back to the channel. It's always beautiful having you here. Today, we're going to spend some time talking about DNA sequencing. Many are times in the life sciences now, we're in the age of molecular biology, molecular science, and we are doing a lot in that sphere. So, you hear sequencing a lot. And the basic question you want to ask yourself is so, what is DNA sequencing? Now, DNA sequencing is any method or the method that is used to determine the composite nucleotides that make up a DNA molecule. So if you have a particular DNA molecule, as a scientist, scientists are interested in looking at the molecule and determining the nucleotide sequence, the nucleotides that make up the molecule one, and then in what order do these nucleotides come. Of course, we know we have adenosine, cytosine, guanine, and thymine. You have these four nucleotides. Now, the question that sequencing wants to answer is these nucleotides, how are they arranged in the DNA molecule? What is the method with which we use to arrange them? Simply that is what we refer to as DNA sequencing. And why is this important? It's important because the sequences of a DNA are the core of life sciences. They are the core of everything that I want to do. They are the core of various characteristics that different type of organisms carry, and so that makes them very important. Now, when you want to do DNA sequencing, there's a workflow. There's a pattern, there's a process, there's I don't want to use the word sequence. There's a workflow to follow, and the first thing to do on the workflow is DNA extraction. Now, what's DNA extraction? You have living organisms, whether they be bacteria, be parasites, be human cells, even plant cells, any living cell, they have DNA inside them. And that DNA, depending on if the cell is eukaryotic or prokaryotic, that DNA will be present either in the nucleus or in the nucleoid region, but they all have DNA. Now, it's important that this DNA can be retrieved. This DNA can be cultivated, can be gotten out of the cell. So this very first process where DNA that is present within the cell of a living organism is harvested, is taken out of that cell and used for the analysis is what we refer to as DNA extraction. And there are many methods of doing DNA extraction, which we will discuss in further videos, but we are taking it one by one. So the DNA that is present in that cell has to be extracted. That is the very, very first process in the DNA sequencing. Now, whether you use kits, there are commercial kits that are available, Gene and different other commercial kits that can be used to do this extraction. Apart from these commercial kits, there are also old methods of boiling. So there are different methods which we'll go into later, but you need to extract the DNA. When the DNA has been extracted, the next thing that needs to be done is fragmentation. The DNA has to be broken down into various segments, into various parts. And this makes it easier to read, makes it easier because it's a very long stretch of molecules with thousands to millions of nucleotides. So, in order to be able to read these sequences, there's a need to fragment the sequences, to break it down into easy to manage, short readable pieces. That's what we call them, the fragments. They are short readable pieces. So, this is important and this occurs. And then we have library preparation. And after the library preparation, in the library preparation, you have the attachment of what we call adapters to different ends of the fragmented DNA. We'll go into details telling us the reason why we do library preparation. After library preparation, then we do the actual sequencing. And the sequencing is where we now begin to determine the actual order in which these nucleotides exist in the fragmented DNA molecule. So, we're able to build back that molecule because we know what the fragments are, and then we can tell you the individual nucleotides that are present in that fragment. And when we have done that, we can now assemble the whole thing as a whole and tell you the nucleotide sequence all through the DNA molecule. That's what is done in DNA sequencing. Now, we'll take our time over a couple of weeks where we'll look at the different processes and what actually goes down in all of these processes. The different technologies, next generation sequencing, nanopore sequencing, and then even Sanger sequencing. We'll look at all of these processes. And then lastly, we look at what are the applications. It's used in genomic, used in medicine, used in biotechnology, DNA sequencing has usage in many areas of science, and it's important that that a life scientist of today, 2026, must understand these concepts and understand what they are used for. Thank you for listening to this video, and remember to like, subscribe, and share, and follow us on this journey as we delve deeper and deeper into the world of science. Ciao.